Hi, it's Dr. Phoenix. Today we're going to be looking at the work of Gregor Mendel, known as the father of genetics. Gregor Mendel was born in Austria in 1822. His education was primarily in the field of physics and math, but then he went on to become a monk in Austria. While he was an Austrian monk, he began to do work with pea plants and discovered some very important and interesting facts about heredity. In fact, Gregor Mendel is now known as the father of heredity because of the things that he discovered back in the mid-1800s. Most of the things that he discovered during his lifetime were not even looked at and considered of any significance. It wasn't until 30 or 40 years after his death that scientists picked up his work and realized what a genius he was, and that the things that he determined were right on, and he was given credit for much of what he discovered during his lifetime, although it was after his death. And many of his findings are now referred to as Mendel's laws. Now, Mendel used pea plants for his studies, and at least part of the reason that he used pea plants is because even though peas are a sexually reproducing plant, and what I mean by that is flowers have a male part that produces pollen, and they have a female part that produces egg. Well, pollen is like the sperm, and of course the sperm fertilizes the egg. In most plants, the pollen is carried by the wind or bees to other plants, and the plants cross-fertilize. But pea flowers tend to stay closed. And as a result of that, most pea plants will self-pollinate. When pollination occurs, we call that fertilization. When a sperm and an egg cell unite, that is called fertilization, and that produces a new cell. We know that gametes are haploid cells, and when they unite and fertilize, they produce a new diploid cell that will ultimately result in a new organism. Okay, so pea plants are self-pollinating, mainly because the flower stays closed. So the pollen of this plant will fertilize the egg of the same plant. And so there really is not going to be any crossing of any genetic material normally. So all of the characteristics that this plant will carry will be only the characteristics that are within the genes of that plant. They're not going to be getting any new characteristics from other plants as a result of self-pollinating. Well, Mendel wanted to produce seeds by joining gametes from two different plants. And so what he did was he took a flower and he cut off the male producing parts, the parts that produce pollen, so that the flower would not self-pollinate. And then he would take a little paintbrush and he would take the pollen off of a different flower and put it onto the egg producing part of the flower where he had cut off the male parts. And so he was mechanically cross-pollinating these flowers. So he would take, for instance, the pollen from a pea plant that had purple flowers and cross-pollinate it with a plant that had white flowers. They would produce peas, which would be the seeds, and once they were planted in the ground, he would see the first generation of plants that came from this crossing. and he would look at their traits. Now, a trait is a specific characteristic that varies from one individual to another. Mendel noticed that in his pea plants there were 7 contrasting traits. For instance, the seeds could be gray and round or white and wrinkled, or they could be yellow or they could be green. The flowers could be white or purple. The pods could be full or constricted, yellow or green. The stem could have flowers within the plant or on the tips of the plants, and the plant could be tall or short. These were 7 opposing characteristics that Mendel was able to observe, and those were the traits that he attempted to cross. Now, the original or the parent plants, in this case these plants here, he referred to as the P generation or the parental generation. So the P generation would be, in this case, a purple flowered plant and a white flowered plant. The offspring of that P generation would be the offspring that were planted in the ground from the seeds that were produced by this plant when the crossing occurred. When these seeds are planted into the ground, that would be the first filial generation, or the F1 generation. This generation would be referred to as hybrids because this would be a crossing between parents with 2 different traits. All right? The parental generation plants were purebred because they were always true breeders. All of these would always produce purple flowered plants. All of these would always produce white flowered plants. But we've crossed the gametes of these 2 plants, and so the F1 generation has gametes from, or has the genes from both plants, and so this is referred to as a hybrid. What Mendel noticed in this F1 generation is that even though there was a crossing of genetic material, the characteristics that resulted in the F1 generation only showed that of one plant. So even though we crossed purple with white, all of the plants turned out purple. The white did not show up at all in the F1 generation. So Mendel concluded that biological inheritance is determined by what he called factors that are passed from one generation to the next. Somehow the purple flowered factors were passed from one generation to the next, and he called these factors, we know now that these factors are genes. So Mendel's factors are today's genes. All right, again, each of the traits that Mendel studied was controlled by one gene that occurred in 2 contrasting forms. All right, one gene that occurred in 2 contrasting forms, tall versus short, yellow versus green, wrinkled versus smooth, and so on. All right. Genes that have different forms are called alleles. All right, so there could be a tall allele and a short allele. All right, it's a gene for height. There could be a gene for pea color, and there are 2 alleles for that, a yellow allele and a green allele. All right, so Mendel came up with 3 major conclusions from his pea plant studies, and these are now known as Mendel's laws. His first conclusion is called the law of dominance. This states that some alleles are dominant and others are recessive. As we just saw, when we crossed the purple flower with the white flower, all of the plants in the F1 generation were purple. That told Mendel that one of these characteristics, in this case the purple flowered plant, was dominant over the other one. And so the dominant gene expresses itself, and the other allele does not. It's sort of hidden. It's not expressed. All right. Mendel's second conclusion from his studies is the law of segregation. After Mendel did his initial crossing with the parental generation and got his F1 generation, he then went and took a F1 generation plant and self-pollinated it to produce an F2 generation. And what he noticed was that even though in the first phyllyl generation there were no recessive traits, in other words, in the purple versus white flowered crossing, there were no white flowers in the F1 generation. When he self-pollinated the F1 generation with themselves, in the F2 generation, the white flowers returned about a quarter of the time, about 25% of the time. So let's summarize. This is our P generation. We cross, for instance, tall with short, and all of the F1 generation plants were tall. That tells us that tall is the dominant trait. Now, if he self-pollinated one of these plants with themselves and created an F2 generation, he noticed that 3/4 of them were tall, but then the short characteristic showed up about a quarter of the time, or 25% of the time. So from this, Mendel assumed that a dominant tall allele masked the corresponding recessive short allele in the F1 generation, but the trait that controlled the short, or the short allele trait, showed up in the F2 plants. So he said that the reappearance of the trait in the F2 generation indicates that at some point the allele for tall had been separated or segregated from the allele for shortness. Okay, that's a mouthful. But remember that Mendel was a mathematician, and he figured all this out based on numbers. He knew nothing about the cell. He knew less about the cell than we do right now as high school students. But from the numbers that he got from experiment, he realized that there must be some form of segregation or separation of these alleles during gamete formation. Now we know now, since we have just studied meiosis, we know that that's exactly what happens. Track with me here. This is meiosis 1, okay. And you may be able to tell that we are looking at metaphase 1 right here. Well, look what happens during metaphase 1. We know that the chromosomes all line up in homologous pairs. So if this is our F1 generation with a tall allele and a short allele, during metaphase 1 they're going to line up side by side, and these two alleles are going to be separated into different cells. They are going to be pulled apart. And so now, by the end of meiosis 2, we're going to see that the allele for tallness and the alleles for shortness have been separated. So we know now that we understand meiosis, that what Mendel discovered by using mathematics from his results is exactly what happens during meiosis. The genes, or the alleles, are separated, and this happens during meiosis 1. So the F1 plant produces two types of gametes, those for the allele for tallness and those for the alleles for shortness, and they will be separated during meiosis. Now, Mendel's third conclusion was that the genes separate independently from one another when they are on different chromosomes. This is referred to as the law of independent assortment. What he's talking about here is that the genes for different characteristics, for instance, this particular chromosome has genes for height, tall and short, but this particular chromosome may have genes for pod color, yellow and green, for instance. Well, what he noticed is that the different traits assort independently from each other, kind of like card shuffling. And so let me point out to you again how this happens through what we now know of as meiosis. Again, we're looking at metaphase 1. And during metaphase 1, we know that the homologous pairs line up in the middle. Well, if there are 4 chromosomes in this cell, they could line up like this, where the big T and the big Y, in other words, the gene for tallness and yellow pods, are on the same side and will end up in the same daughter cell. That is one way they could have lined up. However, they could have lined up like this, where the gene for tallness and the gene for green pods ended up on the same side and therefore ended up in the same daughter cell. So this is a card shuffle. This is totally random how they ended up like this. And so Mendel noticed that there was no order to how different traits express themselves, and he referred to this as the law of independent assortment. Again, his discovery was a result totally of numbers. He had no idea what was going on inside the cell. Now, when organisms have 2 identical alleles, in other words, let's say they have an allele for tallness and an allele for tallness, or an allele for shortness and an allele for shortness, that is referred to as homozygous. Homozygous. Both alleles are the same. Organisms that have 2 different alleles, for instance an allele for tallness and an allele for shortness, that is referred to as heterozygous. All right, so the parents in Mendel's experiments were always homozygous. All right, the parents were big T or little t when he did their crossings. And the F1 generations all ended up like this. These were the hybrids, or heterozygous organisms. Homozygous organisms are true breeding. Okay, if you breed a big T with a big T, they're always going to be tall. If you breed a little t with a little t, they're always going to be short. They're always true breeding. All right, but these heterozygous organisms are the ones where when you cross these, you're going to get some of one characteristic and some of another. Now, when you consider what characteristics an organism is showing, we refer to that as a phenotype. A phenotype is the physical characteristic. In this particular situation, the phenotype of these 3 plants is tall, and the phenotype of this plant is short. So the phenotype is what it looks like, the physical characteristic. All right, that is the phenotype. We can also refer to the genotype, and that is the genetic makeup, or what the genes are in these plants. Well, in order to be tall, there has to at least be one big T. So the genotype for tall could be big T or big T little t. Either one of those would result in tall plants because, remember, the tall allele is dominant. The genotype for the short plant would have to be little t because little t is recessive, and the only way for shortness to express itself is for it to have 2 recessive genes. If there was a big T, then it would be tall. All right. So the terms genotype and phenotype are important terms. Genotype represents the actual alleles that make up the individual, And the phenotype is what is the physical expression of those alleles. I hope that's helpful to you. Please feel free to send me questions on My Big Campus or bring them to class. I look forward to seeing you tomorrow. Have a great day.